Hey, grade 9. This is Miss Cruz to teach you all about genetic cross. But before we start our lesson proper, let's have a simple activity. I have here two primary colors. Now, if we combine red with blue, the result will be a blended color, a secondary color, violet. But what if we combine these two colors? We have an opaque color, we have the color black, and we have a secondary color, violet. Now, what do you think will be the outcome of these two colors? The outcome will be black. This is because black is a dominant color. Therefore, any color combined with black, the result will be black, since it is a strong color. Therefore, it will be more prominent than any other color. So what do you think is the lesson for today? What do you think do this activity imply?
For today's topic, we have Mendelian genetics. But before that, let us know more about Gregor Johann Mendel and his contribution to science. Gregor Johann Mendel is a geneticist who was born in the year 1822 to 1884. He was known as the father of genetics because he is the one responsible for the laws of inheritance of traits, wherein it is stated that traits or characteristics are being passed from one generation to another.
In addition, Gregor Johann Mendel is an Austrian monk. He studied the inheritance of trait using the specimen Pisum sativum, the scientific name of pea plant, therefore developing the laws of inheritance. Between the year 1856 to 1863, Mendel cultivated and tested around 28,000 pea plants. He found out that plants' offspring retain traits of the parents. Gregor Johann Mendel conducted his experiment in a small area in Czech Republic. Mendel stated that traits or characteristics are inherited as particles, but by that time these particles are still unknown. Mendel did not know that the particles were actually chromosomes and DNA. Let us define some terminologies. The first one is genetics. Genetics is the study of heredity and variation. When we say heredity, we are referring to the similar traits that are passed down from parents to offspring or by generation to generation. When we say traits, it refers to any characteristic that can be passed from parents to offspring. For example, we have the color of our eyes, the color of our hair, our skin tone. Next one, we have the gene. A gene is known as the hereditary unit, and also it is just a segment of a DNA. Next, we have the gene locus. A gene locus refers to the certain position of a gene. Summation of all genes, or the totality of all genes in a chromosome, is known as genome. So we have the illustration below. So this is a gene, and summation of all the genes in a chromosome is known as a genome. What is genetics? Genetics is the study of heredity and variation. When we say heredity, we are talking about the similar characteristics that are being inherited by the offspring to their parents. On the other hand, when we say variation, it refers to the differences in terms of trait of siblings and also parent and offspring. What is a gene? A gene is the hereditary unit transmitted from one generation to another.
What is a DNA? DNA stands for deoxyribonucleic acid. This is the genetic material of our body. It serves as the blueprint of our body, since it dictates the color of our eyes, the hair color, the skin tone, and other physical attributes.
On the other hand, a chromatin looks like beads on a string. This is the uncoiled form of chromosome. Lastly, we have the chromosome. A chromosome is a thread-like structure of chromatin fiber and protein found in the nucleus of most living cells, carrying genetic information. To better understand the previous terminologies, let's have this illustration. This double helix structure is known as the DNA. A segment of a DNA is known as a gene. This string-like structure, coiled by nucleosome, is known as the chromatin. During cell division, mitosis and meiosis, this chromatin condense to form a chromosome. Remember that the number of chromosomes differs in terms of species. In humans, we have 46 chromosomes. 23 chromosomes from our mother and 23 chromosomes from our father, with a total of 46 chromosomes. To better understand the topic, let us define more terminologies. The first one is allele. When we say allele, it refers to the two forms of a gene, or the alternative forms of a gene. We have two types of allele. We have dominant allele and recessive allele. Dominant allele is the stronger of the two genes expressed in the hybrid. It represents the dominant trait and can be written in capital letter. Like in our example, we have the uppercase letter R. On the other hand, when we say recessive allele, it is the gene that shows up less often in a cross. It also represents the recessive trait and can be written in lowercase letter. Like in our example, we have lowercase letter r. Genotype is the allele combination for a trait. It is represented by two letters. Like in our examples, the first example is a combination of two uppercase letters or two dominant alleles. The second example is a combination of one uppercase letter or one dominant allele and one lowercase letter or one recessive allele. The third example is a combination of two lowercase letters or two recessive alleles. How about the phenotype? A phenotype is the physical feature resulting from the genotype, meaning to say, this is the trait or the characteristic that can be expressed by interpreting the genotype. For example, we have red and white. We have two types of genotypes. We have homozygous genotype, which is also known as pure because it involves a combination of two dominant alleles or two recessive alleles. Like in our example, we have the first example, which is a combination of two uppercase letters or two dominant alleles. And the second example include two lowercase letters or two recessive alleles. Again, when we say homozygous genotype, it means we have two the same alleles. And if we have two dominant alleles as our genotype, then we can read this as homozygous dominant genotype because it includes dominant alleles. We have two dominant alleles. Again, when we say homozygous genotype, it includes two the same alleles. How about if we have two lowercase letters or two recessive alleles? So how can we read this genotype? So this is called homozygous recessive genotype. Now let's proceed with the second type of genotype. The second type is known as the heterozygous genotype. It is also known as hybrid because it is a combination of different alleles. When we say heterozygous genotype, meaning to say two different alleles. Therefore, it involves one uppercase letter or one dominant allele and one lowercase letter or one recessive allele. So this is called heterozygous genotype. Again, this genotype is called homozygous dominant genotype because it involved two dominant alleles. On the other hand, this genotype is known as homozygous recessive genotype because it involved two lowercase letters or two recessive alleles. The third one is known as the heterozygous genotype because it involved two different alleles, one dominant allele and one recessive allele. To better understand the relationship between genotype and phenotype, let's cite an example. Let's use flower color as our trait. And for the alleles, let's use an uppercase letter R and a lowercase letter r. Let's say that the uppercase letter R or the dominant allele represent the dominant trait, which is red. On the other hand, the lowercase letter r or the recessive allele represent the recessive trait, which is yellow. Now I have here a given genotypes. Now I want you to find the phenotype of the following genotype. Again, when we say phenotype, it refers to the traits or the characteristic that can be expressed by interpreting the given genotype. So we have to interpret the following genotypes. Let's proceed with the first genotype. The first genotype is known as a homozygous dominant genotype. Why homozygous dominant genotype? Because it is a combination of two the same letter, and this letter is an uppercase letter or a dominant allele. So this is known as homozygous dominant genotype. And based on our given, the dominant allele expresses the dominant trait, which is red. So therefore, the phenotype is red. Let's proceed with the second example. The second example is known as a heterozygous genotype. So when we say heterozygous, meaning to say it involves two different letters. We have an uppercase letter R and a lowercase letter r. Based on our given, the uppercase letter R express the dominant trait, which is red. On the other hand, the lowercase letter r express the recessive trait, which is yellow. So what is the phenotype? How come that we have red as our phenotype? This is because the presence of the dominant allele will mask the effect of the recessive allele. Therefore, the trait that will be expressed is red. So red will be prominent than yellow because the dominant allele will mask or will cover the effect of the recessive allele. Let's proceed with the third example. We have a homozygous recessive genotype. Again, when we say homozygous genotype, meaning to say two the same letter, and because it involves two lowercase letters, meaning to say the trait that will be expressed is the recessive trait, which is yellow. Therefore, the phenotype of this genotype is yellow. To understand Gregory Johann Mendel Experiment, let us define the different types of generations that he made use. The first one is the P1 generation or parental generation. The P1 generation is the parent generation. The second one is the F1 generation or first filial generation. When we say filial, it means son or daughter or offspring. It came from breeding individuals from the parent P1 generation. Lastly, we have the F2 generation or the second filial generation. The second generation offspring in a breeding experiment. It came from breeding individuals from the F1 generation. To better understand Gregory Johann Mendel Experiment, let us define the three types of generation that he made use.
The first one is the P1 generation or parental generation. This is the parent generation. Based on Mendel experiment, he made use of the pure genotypes for this generation. Moving forward, let us now discuss Mendel's pea plant experiments. Before we proceed, let us answer this question. Why did Mendel use peas or Pisum sativum in his experiment? The first reason is that it can grow in a small area. Remember that Mendel conducted his experiment in a small garden in Czech Republic. Second, it can produce lots of offspring. Mendel cultivated around 28,000 pea plants in a short period of time. Third one, it can either be self-pollinated or cross-pollinated, meaning to say, Mendel can easily manipulate the expression of traits. These are the 7 traits that Mendel subjected in his experiment. We have seed shape, seed color, pod color, plant height, flower position, and flower color. For the seed shape, the dominant trait is round, the dominant allele is uppercase letter R. The recessive trait is wrinkled, the recessive allele is lowercase letter r. For the seed color, the dominant trait is yellow, the dominant allele is uppercase letter Y. The recessive trait is green, the recessive allele is lowercase letter y. In pod shape, the dominant trait is smooth, the dominant allele is uppercase letter S. The recessive trait is wrinkled, the recessive allele is lowercase letter s. In pod color, the dominant trait is green, the dominant allele is uppercase letter G. The recessive trait is yellow, the recessive allele is lowercase letter g. In plant height, the dominant trait is tall, the dominant allele is uppercase letter P. The recessive trait is short, the recessive allele is lowercase letter t. In flower position, the dominant trait is eggshell, the dominant allele is uppercase letter A. The recessive trait is terminal, the recessive allele is lowercase letter a. In flower color, the dominant trait is purple, the dominant allele is uppercase letter P. The recessive trait is white, the recessive allele is lowercase letter p. Proceeding, I have here the illustration of the characteristics that Mendel observed in his experiment. We have flower color, flower position, seed color, seed shape, pod color, and stem length. In this slide, you can see the yielded result from Mendel pea plant experiment. In the column 1, you can see the traits of the parental generation. Again, when we say P1 generation, it stands for parental generation or the parent generation. And as we said in our previous slide, the parent generation have a pure genotype. That's why in this column, both the dominant trait and the recessive trait were expressed. Now, let's proceed with the next column, the F1 generation or the filial 1 generation. In the F1 generation, actually F1 generation is the offspring of the P1 generation, and based in the yielded result, only the dominant trait were actually expressed. This is because the F1 generation have a heterozygous genotype. And when we say heterozygous, meaning to say it involves 2 different alleles, one dominant allele and one recessive allele. Dominant allele will cover or mask the effect of the recessive allele, therefore making the dominant trait more prominent. Now let's proceed with the third column. We have the F2 generation. The F2 generation is the offspring of the F1 generation. And based in the F2 generation, both the dominant trait and the recessive trait were actually expressed with approximately a ratio of 3:1. Before we move forward, I have here a question. Did the observed ratio match the theoretical ratio? The theoretical or expected ratio of plants producing round or wrinkled seeds is 3 round is to 1 wrinkled. Mendel yielded a result which is 2.96 is to 1. The discrepancy is due to the statistical error, meaning to say, the larger the sample, the more nearly the results approximate to the theoretical ratio. So what do the peas looks like? So I have here a picture of a pad. So this pad houses the peas. So we have two types of peas. We have wrinkled and we have smooth. When we say pea plant, the scientific name is Pisum sativum. When we say Pisum, it is the genera. When we say sativum, it is the species. So we actually name a living organism based on a phylogenetic tree. Let's discuss Mendel's laws of inheritance. The first law of inheritance of trait is known as the law of dominance, where it is stated that in a cross of parents that are pure for contrasting traits, only one form of the trait will appear in the next generation. Meaning to say, all the offspring will be heterozygous and express only the dominant trait, like in our given example. So we have the genotype of parent 1, which is a homozygous dominant genotype. So it includes two dominant alleles, cross with the genotype of parent 2, which include two recessive alleles. Now, if you cross the genotype of parent 1 with the genotype of parent 2, the yielded result will be a heterozygous genotype. So when we say heterozygous genotype, it includes one dominant allele and one recessive allele. And always remember that the presence of the dominant allele will mask the effect of the recessive allele. Therefore, the prominent trait will be the dominant trait, which is round. To better understand the law of dominance, let us have this illustration. Based on the given picture, we cross parents who have both a heterozygous genotype. Now, what will be the genotypic ratio? So we will have one homozygous dominant genotype is to 2 heterozygous genotype is to 1 homozygous recessive genotype. What will be the phenotypic ratio? The phenotypic ratio will be 3 yellow is to 1 green. So how come we have 3 yellow? So we have 3 yellow because always remember that the heterozygous genotype express dominant trait as well as the homozygous dominant genotype. Let's proceed with the second law of inheritance of trait, which is the law of segregation, wherein it is stated that during the formation of gametes, egg or sperm, the two alleles responsible for a trait separate from each other. Alleles for a trait are then recombined at fertilization, producing the genotype for the traits of the offspring. Let's apply the law of segregation in this picture. So we will use uppercase letter T to represent the dominant trait and lowercase letter t
to represent the recessive trait. Now, based on the law of segregation, the alleles in the gamete of female parent segregate during fertilization. The same goes with the male parent. It separates. Then after that, it recombined. So we have the combination of the genotype of parent 1 and parent 2 inside the boxes. So the letters in the boxes represent the genotype, the possible genotype of their offsprings.
Let's proceed with the last law of inheritance of trait, known as the law of independent assortment, wherein it is stated that alleles for different traits are distributed to sex cell and offspring independently, meaning to say the inheritance of the hair color is independent to the inheritance of the eye color.